Hello everyone, how are you? So this lecture we will be discussing isothiocyanate glycoside. So before we start about this lecture, we'll go through the learning outcomes. At the end of this lecture, you will be able to define isothiocyanate glycoside, okay? And also you'll be able to outline natural occurrence of isothiocyanate glycoside with few examples, and you will be able to explain the botanical name, family name, chemistry, and uses of only two species, that is black mustard and white mustard. So in the whole chapter, mainly we will be covering two examples of plant species that contain isothiocyanate glycoside. So before I discuss about isothiocyanate glycoside, I would like to show you what is the isothiocyanate functional group. Let us have a look. So when it is NCO, this is known as isocyanate. But instead of oxygen, when we replace it with sulfur, then we call it as isothiocyanate. So when we talk about isothiocyanate glycoside, that means these are the glycosides which contains a glycon part having this thiocyanate functional group. So thiocyanate containing functional groups in glycon, they are known as thiocyanates, and when it is attached with sugar molecule, they are known as isothiocyanate glycosides. Coming back to the lecture again, they are also known as thiocyanate or sulfurated glycosides. And here, as I mentioned already, glycons are thiocyanate in nature, and they are also known as glucosinolate. This is again one of the new example that we are going to discuss. Okay, glucosinolate means these are class of naturally occurring compounds which contains sulfur and nitrogen, and they are mainly derivatives from the derivatives of sugar and amino acids. Okay. So when we talk about glucosinolates, the isothiocyanate glycosides are also considered as a class of glucosinolate compound. So glucosinolate, in their structure, they contain a central carbon atom which is attached to a sugar via sulfur atom, whereas this central carbon atom is also attached to a sulfate group via nitrogen atom. And this central carbon also contains a R group which can be different from one glucosinolate to another glucosinolate, and it is depending upon the R group, the activity of different glucosinolate differs from each other. Let us have a look at the structure. So as I mentioned, glucosinolate contains a central carbon atom which is attached to nitrogen in one side, and other side it is attached to sulfur. So glucosinolate, you can see again, it contains a isothiocyanate system, and via sulfur it is attached to sugar, and via nitrogen atom it is attached to sulfate group. So any compound, any naturally occurring compound which contains this system, okay, they are known as glucosinolate compound. And why isothiocyanate glycosides are classified under glucosinolates? Because they also contain the SCN system which is connected to sulfate group as well as sugar moiety. We will see the examples, then we will have a better understanding. They are mainly There means the isothiocyanate glycosides, They are mainly abundant in families like Brassicaceae, which is also known as crucifery, and Capridiaceae. And it is believed that these glycosides provide plants, or protect plants from the various parasites, or against various parasites as a plant defense mechanism. Now, regarding the biosynthesis, there are two biosynthesis routes. One of them is shikimic acid, which acts as a precursor for the synthesis of aromatic glycoside, that is aromatic isothiocyanate glycosides. One of the example is cinalbine, which is mainly available in the seeds of white mustard. And if you look at the structure of cinalbine again, it is an example of isothiocyanate glycoside, as well as it can be classified as one of the type of glucosinolate. If you look at the structure, it has a central carbon atom which is attached to a sulfate group by a nitrogen, and which is also attached to a glucose by a sulfur, and it also contains the SCN system with the isocyanate. So it is an example of isothiocyanate glycoside because it is attached to a sugar molecule. Second example is which is derived from acetate units, which act as a precursor for the synthesis of aliphatic glycoside. So you look at the difference. The example here is cinegrine, which is mainly available in black mustard seeds. And if you look at the difference, the acetate units act as a precursor for aliphatic isothiocyanate glycoside, whereas shikimic acid acts as a precursor for the synthesis or biosynthesis of aromatic glycoside like cinalbine and cinegrine, which is available. If you look at the difference between cinalbine and cinegrine. Now, this is a picture of black mustard seed. Nothing new about it. I'm sure all of you know about it, and you have been consuming it in the preparation of the different curries, spices. And this is how the flowers of the plant of black mustard looks like. Its botanical name is Brassica nigra, and family name is Brassicaceae. And as it can be understood, the used part of this plant is mainly the black mustard seed. That is this picture, seeds of the black mustard. Talking about the chemistry of black mustard, the seeds of black mustard contain active principle, that is cinegrine, which is also known as potassium myrinate. And hydrolysis of the cinegrine, hydrolysis of cinegrine produces allyl isothiocyanate, which is nothing but the constituent of mustard oil. It also contains, that means the black mustard seed, other than cinegrine, it also contains various fixed oils, proteins, as well as mucilage. Regarding the uses, the black mustard seeds are mainly used as stimulant, which temporarily quickens various vital processes of the human body. It is also used as emetic in excess quantity, to get rid of any poisoning of the GI tract. And it is also found to be useful as a diuretic, which increases the urination. And most common use of black mustard seed, of course, as a condiment, that's a preparation of spice to enhance the flavor or taste of any food. And another important medicinal use of black mustard seed is they are also used as rubefacient. What's the meaning of rubefacient? Rubefacient, they are the substances which causes redness of the skin, by dilation of capillaries and also by increasing the blood pressure. And thus they are mainly useful to relieve acute or chronic pain. So you can see black mustard seeds are mainly biologically used, as rubefacient, as diuretic, emetic, as well as stimulant, and it is also used as condiment to enhance the flavor or taste of food. Now coming to the next variety, that is white mustard seed. Here the botanical name is Brassica alba or Sinapis alba. You can look at the white mustard seed picture, how it looks like. It also belongs to the same family, that is Brassicaceae. And used part, obviously, the medicinal used part is mainly the dried seed. Even though the leaves of this plant are also consumed as vegetable, okay, which has some benefit, but here mainly we are concentrating about the used part seed, which contains the biologically active principles. Now talking about chemistry, the seeds of white mustard, it contains the active principle, that is synalbine, and the hydrolysis of synalbine produces the glycon part of the glycoside, that is acrynyl isothiocyanate. It is the structure of acrynyl thiocyanate plus glucose. Other than synalbine, the white mustard seed also contains fixed oil around 30%, starch content around 4%, mucilage, that is the epidermis of the seed coat. Not only that, they also contain proteids around 25%. What are proteids? Proteids are nothing but the food substances that contain nitrogens. So other than synalbine, they also contain fixed oil, proteids, starch and mucilage. Now talking about the uses of white mustard seeds are almost similar to the black mustard seed. They are used as stimulant, emetic, also as a diuretic agent, and as a diuretic they can be useful to reduce the blood pressure of a patient, and also used as condiment like black mustard, also used as rubefacient just like black mustard, and moreover they are found to be useful as counter irritant to relieve irritation of the skin. Now these are the two examples mainly, that is the black mustard and white mustard seeds which represents the isothiocyanate containing glycosides. Now let us have a look at certain questions. Let me check your knowledge. Which of the following glycoside is present in black mustard seed? Synalbine, salicin, synegrine, sapogenine. The correct answer is synegrine. I would like to mention here, these two names are a bit similar, synegrine and synalbine. How to remember which one present where? Remember synegrine, negrine from the negro. Of course, I am not a racist, just we know that negro also represent the beautiful color black. So synegrine, negrine from negro, you remember, from the synegrine belongs to black mustard seed. Next question, which of the following a glycon is obtained after the hydrolysis of synalbine by the enzyme myrosinase? Option A, glycyrrhizinic acid, glucuronic acid, allyl isothiocyanate, acrynyl isothiocyanate. The correct answer is acrynyl isothiocyanate. Next question, which is the base of this, the basic question of this chapter. Which of the following is an isothiocyanate glycoside? So look at this four structure. Structure A, no. Structure A, if you look at the structure carefully, it contains a CN group and plus sugar. That means it is an example of cyanogenic glycoside, not isothiocyanate. Then what is the correct structure, correct answer? Answer B, no. Even though, if you look at the structure, even though the structure contains isothiocyanate group, that is NCS, it is not an isothiocyanate glycoside, because the question is asking which one of the, this is an example of isothiocyanate glycoside, because it is not attached to a sugar moiety. So it is just an example, structure B is just an example of a glycon containing isothiocyanate. So the correct answer is D, option D. Why D? Because if you look at the structure, it contains isothiocyanate, okay, the SCN, which is attached to a sugar moiety. So this is an example of isothiocyanate glycoside. glycoside means it must be attached to a sugar moiety. So always remember this, especially when you go for exam, you tend to forget that glycoside means it must be attached to a sugar moiety. Not just only look for the functional group NCS, also look for whether the whole molecule is attached to a sugar molecule or not. Okay. So with this, I finish my lecture, and these are some of the reference textbook. Okay. Other than textbook, you can also look for the information in supplementary books. And thank you for your attention. See you in the next class again.